Hello, everyone. Thank you for joining our webinar. I'm Haley from RWD Life Science. We noticed from your registration details that many of you are joining from different regions, including Korea, Japan, China, and Italy. So let me say hello in a few languages. Konnichiwa, welcome our friends from Japan. And annyeonghaseyo, welcome our friends from Korea. 大家好. Welcome our friends from China. And of course, hello to everyone joining in English. So today, I'm very pleased to officially introduce our newly launched RWD Lab Academy series. This series is a little bit different from our previous RWD Academy, which mainly focus on literature reading and sharing recent research advances. So RWD Lab Academy is more practice-oriented and all focused on hands-on technical content. We will hold online sessions from time to time, covering topics such as the experimental techniques, troubleshooting, and standardized workflows. So the topic of today's webinar is fiber photometry step-by-step along the experimental workflow and practice tips. So in this webinar, we will focus on experimental preparation and surgery, data acquisition, and data analysis. During the presentation, you are very welcome to join the discussion and submit your questions in the question box. And in the final Q&A section, we will select some questions to answer. We will also receive many questions during registration, and we will choose some of them to address as well. So if your question is selected, you will receive a small gift from us, such as ceramic cannula. So let's begin our today's presentation. So first, let us briefly look at the basic principle of fiber photometry. Fiber photometry records brain activity by measuring fluorescence changes through an implanted optic fiber. When the neurons are active, the calcium ions enters the cells and the calcium indicators become brighter. So these fluorescence changes can be used to reflect neural activity. And in addition, they are also sensors for neurotransmitters, such as dopamine and glutamate. When these sensors bind to specific molecules, the fluorescence changes, allowing us to monitor changes in neurotransmitter levels. At present, fiber photometry is widely used in many research areas, including the learning and memory, reward and addiction, emotion and pain and motivation, and so on. Fiber photometry also has many practical advantages over other methods for in vivo monitoring of neural signal in animals. For example, compared with the electrophysiology, fiber photometry provides better cellular and molecular specificity. And compared with FSCV, fiber photometry can offer higher sensitivity and more specific detection in some applications. So for example, when we're measuring dopamine, FSCV can't always clearly distinguish dopamine from other structurally similar neurotransmitters, such as the norepinephrine. And compare with the microdialysis, which has a relatively slow sampling rate, usually with intervals longer than 1 minute and often around 10 minutes. Fiber photometry provides both higher spatial resolution and much higher temporal resolution. And compared with the miniscope and two-photon imaging, fiber photometry is much less invasive surgically, places fewer constraints on lateral animal behavior, and is lower in cost and complexity. It is relatively easy to use, supports plug-and-play operation, and offers good reproducibility. So we can see from this the advantages of fiber photometry from these figures. Based on the principle of fiber photometry, the system needs an implanted optic fiber to deliver the excitation light and collect fluorescent signal from the target brain region. So for this reason, a stereotactic surgery is usually required before the recording experiment. In most cases, the surgery is also used to deliver the virus sensor in the target brain region. So before starting the surgical procedure, we need to prepare the following instruments and consumables. For example, the anesthesia machine, the stereotactic instrument, and the micropipette puller, and also the injection pump. So before a fiber photometry experiment can begin, the stereotactic injection is generally required to deliver the virus into the target brain region. The main steps usually include the anesthesia, the animal fixation, hair removal, skull leveling, and virus injection, then cannula implantation, and dental semen fixation. So next, I will briefly go through the surgical procedure. So we start with the anesthesia and fixation. So first, with the mouse and induce anesthesia with isoflurane in the induction chamber. And after that, we maintain anesthesia with a mask and shave the head and face the mouse in the stereotactic frame. So during the surgery, make sure the head is firmly fixed by gently moving the tail side to side. And you can regularly check the depth of anesthesia by the tone pinch. So, and before the incision, we need to clean the scalp and apply the erythromycin eye ointment to protect the eyes. And after fixation, make a midline incision and fully expose the skull. And we clean the skull surface. And next, we can move on the skull leveling. The purpose of this leveling is to make sure the skull is level, so the cordless will be accurate and the injection or implantation can be done in the correct brain region. So then we bring the electrode to the bregma and set it as the reference point and arranging. And then compare the height difference between the bregma and lambda in the arterial and posterior direction. If the difference is higher than 0.03 millimeter, we adjust the loose clamp. Then after that, we compare the skull height on the left and right sides, usually 2.3 millimeter from bregma. Yeah. So now we move to the left side, and then we move to the right side. If the difference is higher than 0.03 millimeter, we adjust the ear bars. After any adjustment, the bregma should be reset as the origin. So once the skull is leveled, we use a micro drill to make a small hole in the skull, and then we move the electrode above the target site and lower it to the target depth, and we set the injection parameters, and we start the viral injection. And here are a few important notes to keep in mind. So we lower the electrode slowly and at a constant speed. And after the injection, please leave the electrode in the place for about 10 to 15 minutes. So there's an important practical question is how to choose the appropriate virus. So when we're choosing a calcium sensor, there's no single best option for everything. The most important question is, what do you want to measure? So first, we think about speed. The fast processes like neural firing need fast sensors, while slower processes can use more sensitive but slower ones. And second, we consider the sensitivity and location. The calcium level vary across the cell. If the calcium levels are high, you want a low affinity sensor, so it doesn't get saturated. If changes are small, you want a more sensitive sensor. And also, some sensors, they designed to work in specific parts of the cell, like nucleus, ER, or membrane. So you need a sensor that matches the range and works in the right compartment. And finally, we think about the multiplexing. So if you also want to measure other signals like neurotransmitters, such as dopamine. You may need sensors with different colors to avoid overlap. So in short, it comes down to the speed, sensitivity, and the compatibility with your experiment. A simple way is to check the website of viral vector providers, such as the Allgene, where you can find basic information related to fiber photometry indicators, including the introduction to the different indicators or probe tools, the labs they come from, and the corresponding publications. And actually, you can also consult your local viral vector companion for more detailed support. In addition to calcium ions, fiber photometry can also be used to monitor real-time changes in neurotransmitters. Many neurotransmitter sensors have now been developed, such as the probes for dopamine, the norepinephrine, and the serotonin, and so on. Those tools are widely used in the research on emotion, sleeping, learning, and memory. The Professor Li Yulong Laboratory at Peking University is a benchmark and a leader in the development of the neurotransmitter probe tools in the industry. So their lab has its own website where they regularly update the newly developed neurotransmitter probe tools and the corresponding published articles. So you can find frequently and ask questions and answers about the probes on the website. After the viral injection, the last step is the fiber implantation. Before implantation, choose a suitable fiber optic cannula. Most cannula on market consist of two parts, the ceramic cannula and the optic fiber. Different models can be selected based on the experimental needs. The main parameters to consider include the core diameter, numerical aperture, and the length, and also the outer diameter. So the next question is, what parameters can we choose, and how do we choose them? And here are some available parameters to choose from. So the material, the type, the color, white or black, the core diameter and outer diameter. And how do we choose them? So the conventional recommendations are as follows. So for mice, 100, 200 micrometer core diameter, 0.22, 0.39 numerical aperture, and 1.25 millimeter outer diameter. And this is for the rat. So now after the cannula is placed at the target depth, it needs to be fixed with dental semen. So there is a common practical question is, how can we prevent the optic fiber cannula from falling off? So there are several useful tips. So first, we clean the skull surface before applying the dental semen. And we need to remove the connective tissue, bone fragments, and the saline, and make sure the skull is completely dry. And second, just like this picture, we drill slowly and gently roughen the skull surface without drilling through it, so that the semen has a larger contact area. And third, tissue adhesive can also be considered. Here I would like to add one important reminder. If you plan to record from multiple brain regions at the same time, it is recommended to finish all the viral injections first, and then we implant all cannula together. The reason is simple, because the mouse brain is small, and once one cannula is implanted and fixed with dental cement, the cement can easily cover the bregma point. So if the bregma is no longer visible as the reference point, it becomes more difficult to accurately locate the other target regions, which may affect the accuracy of the experiment. So now up to this point, the surgical procedure is complete. After surgery, we return the mouse to its home cage and allow the virus to express for about 2 to 3 weeks before starting the fiber photometry experiment. So during this period, the animal's condition should be monitored continuously. At this point, a common question is, does fiber photometry always require injection of a fluorescent protein virus? Yeah, the answer is not necessary. The most common approach is to inject a fluorescent viral vector with a specific promoter to label the target cells. But transgenic animals can also be used to express fluorescence proteins. So for example, commercially available chicken mice, yeah, it doesn't require fluorescent virus injection. About 2 to 3 weeks, the animal is ready for the fiber photometry experiment. During the signal acquisition, the excitation wavelength should be selected according to the indicator or sensor used. In general, 410 nanometer is commonly used as a control channel. 4670 nanometer is used to excite green fluorescent signals, and 560 nanometer is used to excite red fluorescence signals. And in addition, our full fiber photometry system also includes 640 nanometer excitation, which is used for the far red fluorescent signals. So we can provide a various model of fiber photometry system, like the dual color, tricolor, and the four color. And our system is equipped with 2 built-in CMOS cameras, and is also compatible with the same site, the optogenetic stimulation. In addition to choose the right excitation wavelengths, the consumable selection is also very important. We also need to prepare the low autofluorescence optic fiber, the sleeve, and the related accessories. There are 2 key points to keep in mind. So first, the parameter of the optic fiber and sleeve should match the implanted cannula, including the core diameter and NA. Otherwise, the signal transmission efficiency will be affected. Second, the diameter of the fiber end and the sleeve should also match the outer diameter of the ceramic cannula. Otherwise, the optic fiber cannot be properly connected to the implanted cannula through the sleeve, just like this. So if you record a multiple brain regions, like in this imaging, or from multiple mice at the same time, then 1 to multiple fibers should be selected. Physically, each fiber is still independent. They are only bundled together side by side at the combined end. In the software, they are also displayed as the separate spots, and the signal detected from each fiber remain independent. This is why signals from different brain regions or different mice don't mix together. Yeah. And for long time recording or experiments in which the animal moves a lot, a rotary joint can also be used. However, there are 2 points to consider. So first, in fiber photometry, both excitation light and the return fluorescent signals are relatively weak. So usually only tens of microvolts. So the rotary joint may cause signal loss and affect the weak signal detection. And second, friction during the rotation may induce some additional noise. So in practice, the benefit of using a rotary joint should be balanced against this potential impact on the signal quality. Yeah. So after preparing the required consumables, the next step is connect the system. The connection starts from the main unit. then to the fiber. And if a rotary joint is needed, connect the rotary joint first, and then connect the fiber. Now we can move on to the signal acquisition. Okay. Yeah, so because of the presentation, maybe it doesn't work, I will play a video to tell us how to set the light setting. Just wait a second. Yeah, thanks for your patience. So we can continue. Yeah, so this one is the signal light setting. The first step is the light source setting. So now we will set the 410 nanometer and the 470 nanometer. So this step is to match the excitation wavelengths to the indicator or the sensor we use. For example, if Gcam was injected, we select the 410 and 470. Yeah. If the dopamine sensor was also injected, we will select the 560. Yeah. And here are some practical tips. So we can see from here. So before the experiment, please test the output power at the fiber end to ensure it is within a suitable range. And we place the fiber end on the power meter for measurement. And for the mouse experiments, 20 to 50 microwatt is generally recommended for the 470 nanometer and 560 nanometer. And in previous studies, around 30 microwatt is commonly used in mice. And for rat, yeah, there are some parameters for rat. Yeah. Actually, in practice, the final setting should be determined based on the animal species, sensor type, and the pilot testing. Next, we will move to the channel setup. Yeah. So we will set the channel to make sure the signal is clearly captured from the fiber end. So first we set the frame rate from 10 to 215 FPS, and 60 to 100 FPS is commonly recommended. Then we set the exposure time. After that, we gently move the fiber end toward light source and increase the gain, and we check the end. And next, we will select some channels from channel 1 to channel 9, and we also will adjust the ROI so that it will stay within the fiber end. Yeah. And finally, we will reduce the gain back to 1 and save the settings. After all the settings are ready, the next step is to connect the optic fiber to the implanted cannula using the sleeve. Then, based on the experimental design, we choose how to add the event markers. Event marking is important because it allows the fluorescent signal to be aligned with the stimulation or behavior during later analysis. So there are two common ways to add markers. So the first one is automatic marking through TTL signals. For example, when the photometry system is connected to the external devices, such as an optogenetic stimulator or a field conditioning chamber. And second is the manual marking based on the behavioral events observed in the video. In many fiber photometry experiments, the neural recording is performed together with the animal behavior testing, such as the open field test or the activated plasmids. This makes it possible to relate the fluorescent changes to the animal's behavior in real time. So The common paradigms used to evolve signals include the electrical stimulation, optogenetic stimulation, the chemogenetic stimulation, and some behavior events. Yeah. So these event markers provide the reference point needed to link neural signals with stimulation timing or specific behaviors. Yeah. So to do this, we can collect animal behavior camera so that the behavior video is recorded at the same time as fluorescent signal. And the software can display trajectory tracking. In addition, when the animal enters to the ROI, the system can also perform the automatic ROI-based event marking. Yeah, you can see in this video. And in this example, we collect 3 behavior cameras at the same time, all connected to the cameras, support the trajectory, tracking, and the ROI event marking. Yeah. So you can see these different colors is the different event markers. Okay. So all the parameters have already been set, so we can now start recording. Yeah. So next, I will use our software to give a demonstration. And this is a video that we record in the morning. Yeah. So we have already we inject the NE sensor virus in the LH area in the mouse, and the stimulations paradigm is the tail suspension. So now all the parameters have been already set. So now we can start recording. This is for preview, and this is for the recording. So we click here. So you can see the baseline over here. And when we began the tail suspension, so maybe you will see the 470 nanometer fluorescent signal is increasing. You can see here, which means the fiber photometry system is detecting the signals. Yeah. So now we place the mouse into the cage, and you can see the signal is gradually decreasing. So a common question at this point is, what if instruments or, sorry. So there is a common question is, there's no signal detected in the fiber photometry. So mostly it's because of the collection, the virus, or the parameters, or the instruments. So for example, the virus, sometimes because of the poor virus expression, and sometimes you have good expression, but the cannula out of the virus area is not at the target area. And also sometimes the normal, it is normal because no signal might be the actual situation. So before event analysis, fiber photometry data usually need to be pre-processed to reduce noise in the signal. Common pre-processing steps including filtering, bleaching correction or baseline correction, and motion correction. In most experiments, the meaningful physiological signal is mainly located in the low frequency range, while the noise is distributed across all frequencies. So for this reason, the data can be processed with a low-pass filter or a moving average algorithm to reduce the high-frequency noise and improve the signal-to-noise ratio. So in RWD software, the smoothing process using a moving average algorithm to smooth the data and reduce the sawtooth like fluctuations. The smoothing parameters can be set from 5 to 50. A higher value produces a smoother trace. Another common issue is photobleaching. When fluorescence of calcium indicator is exposed to light for a long time, bleaching can occur. This reduces both the baseline level of the signal and the amplitude of signal changes. To correct the gradual downward trend caused by the photobleaching, appropriate baseline correction algorithms can be applied. So in our software, baseline correction can be selected to compensate for the downward drift in fluorescence during long-term recording, including effects from photobleaching and LED thermal effects. 3 baseline correction methods are available. Yeah. When animals move freely, the change in light transmission may cause by the activities such as movement, turning or jumping the wire, generating motion noise. There are usually 2 ways to correct the motion noise. The first one is the co-expressed indicator with a control fluorescence group, for example, red fluorescence protein. Or the second is detect the isospastic point signal of the indicator. The fluorescence of the calcium indicator will change with the change of calcium iron concentration, while the signal collected at isospastic point will only remain within a relatively stable range. So that is, the collective fluorescence has nothing to do with the physiological signal, and then the motion noise is processed and corrected through an algorithm. So our software includes a built-in motion correction algorithm for this type of pre-processing. After pre-processing, the last step is event analysis. In most cases, the start time of event is set as the time 0, and the fluorescence changes before and after the event are analyzed. The 2 most common used readouts are delta F over F and the z-score. Delta F over F represents the change in fluorescence relative to the control, or in simple terms, the relative increase in signal compared with the control. It tells us how much the signal has changed. This score can be understood more simply as how far the signal is from the average value. Based on delta F, I further standardized the signal using the standard deviation and shows how many standard deviations the current signal is above or below the mean. In other words, delta F over F shows the size of signal change, while the z-score shows how strong or significant that change is relative to control variability. And both delta F over F and z-score are widely used in fiber photometry studies and are commonly reported in the literature. Another commonly used quantitative index is AUC, or area on the curve. It measures the area enclosed by the curve and a reference Y value, and can be used to evaluate the overall magnitude of fluorescence change. A fluorescence heat map is also often shown together with delta F over F and AUC. The brighter region in the heat map indicates stronger neural activity, making it easier to visualize the distribution of signal changes over time. Another useful group of metrics is peak analysis, including the peak quantity, frequency, and peak mean value. The peak quantity refers to the number of detected events within a given time period and reflects how many times neural activity occurred. And peak frequency is the peak quantity divided by time, representing the rate of activity. Peak mean value is the average height of all detected peaks, reflecting the average strength of each event. For example, this study compared average peak delta F over F calcium event frequency when the mice were exposed to a home cage versus an enriched or novel environment. In many studies, fiber photometry is combined with behavior assay to investigate neural activity during free movement. In experiments such as open field test, elevated plasmids, it is often important to analyze fluorescent signals in relation to the animal's location. So RWD's new fluorescence place heat map feature visualize the relationship between the behavior position and the fluorescence intensity. So last, let me briefly show how the data can be analyzed in the software. So now we imported the data. After data import, the waveforms can be displayed over here. In the overview channel, you can also see the basic information, including the light source, the frame rate, the channels, and recording durations. The last step is data pre-processing. Here we first select the signal to be analyzed, and then we choose the control. So we choose 470 and 410. The software supports several common pre-processing functions, including smoothing, baseline correction, and motion correction. And just wait a second, we can change some parameters to make this curve better to see. So we can change like this. Yeah, I will click plot. Yeah. So yeah, by clicking the plot, we can generate the delta F over F trace. Yeah. It can also perform the automatic peak analysis. You can define the statistic duration, the MAD duration, and the amplitude threshold. Here, the MAD means absolute deviation. In simple terms, the software uses a median-based baseline, and only signals above the selected threshold are identified as peaks. So you can also set a minimum interval between the peaks and choose the analysis channel and display the peaks results directly on the trace. And after that, the software can automatically calculate the parameters such as peak quantity, frequency, and mean value. After the pre-processing, we move to event analysis. All event markers can be displayed in the software. So you can see these markers. And when the video is played, or when the signal timeline is moved freely, The signal and the video remain synchronized, and event marker can also be added or deleted manually. So for example, during manual marking, you can correct the event based on a specific animal behavior seen in the video, such as start of tail suspension. Yeah. Once the event markers are selected, you can define the time duration before and after each event. Then with one click, the software can generate both fluorescence heat map and curve. Up to this point, we have been processing and analyzing data from a single mice. If we want to compare different groups later, we can click heat map. So now I will input 3 data. And here we are showing 3 example groups, but in practice, each group can include data from multiple animals. And we click. The software can generate both a fluorescence heat map and a curve, and you can view data from more events, individual animals, or the average of the group. The software can also calculate the AUC. Yeah, we click here. Then you can see this result. Yeah. So after that, the data can be combined for the statistical analysis. So the next is the trajectory analysis. The software can automatically generate a trajectory heat map and the trajectory trace plot by opening the ROI. You can see here. You can view the ROIs defined during the recording as well as the time points when the animal entered to each ROI and duration spent in each region. And this is the fluorescence place heat map. So now we import the fluorescence data and trajectory data. Here you can see in which locations the mouse shows stronger fluorescence activity. All the figures shown above can be saved and used directly. So this is the end of the analysis. Let's move back to our presentation. Yeah, so before we move on to the final part, our Q&A part, I would like to briefly introduce our overall solution. At RWD, we provide one-stop solution for fiber photometry experiments. This includes not only the recording system, but also some related instruments and consumables we mentioned today. The fiber photometry offers clear advantages for in vivo neural recording, including high sensitivity, good specificity, and flexible integration, and relatively low complexity, make it a practical and reliable tool for many research scenarios. So for example, the prop development, detection, the neural circuit research. As you can see on this slide, our system has already been widely used in more than 600 laboratories worldwide, including many leading universities and the research institutions. We also offer a free trial program. So if you are interested in, so we are very welcome you to scan the QR code or contact us for more information. Yeah. So As we come to the end of today's webinar, we'd like to take a short moment for Q&A session. So during the webinar, we've received a number of questions in the chat. Yeah, we also collected many questions during registration. And thank you for all your active participation. And now let's first answer some questions we collected during registration. And the rest of you may continue to submit your questions in the Q&A box during this session. Yeah. First, where is the optimal implantation position for the ceramic cannula? And second is, what is the recommended length for the ceramic cannula? Yeah. So for fiber photometry experiments, the recommended implantation position is 50 to 100 micrometer above the viral injection site, yeah, for best results. If the distance is too large, you can see from this picture, the detected fluorescent signal may decrease, but calcium indicators and neurotransmitter sensors have gone through many iterations and with new virus. Yeah, so a slightly larger offset is often still acceptable. It is generally recommended to run a small pilot experiment first to optimize the setup. Yeah. So the second question, so we recommend you the length should be 1 to 2 millimeter longer than the depth of the target brain region, because we need living room for the skull thickness, you can see here, and for some fixation materials such as dental cement. Yeah. And in fiber photometry experiments, there are some artificially introduced that can affect results. These include the autofluorescence caused by the tissue damage or motion-related signal changes when animals move during free behavior and photobleaching for long-term imaging. So to help deal with this issue, we use 410 nanometer as an isospastic point. At 410 nanometer, the signal is not sensitive to the, you can see here, to the fluorescent protein itself. Instead, it mainly reflects the interference vectors and background noise. So the 410 nanometer signal can be used as a reference for background correction. Yeah. The next question is, how does the data analysis synchronize with EEG or optogenetic recording signals? So for optogenetics, when the system delivers light stimulation, a TTL signal is sent to the fiber photometry recording system to automatically generate timestamps. For example, this is one of our customers' data. So you can see these events. Yeah. For EEG signal synchronization, our system supports importing EEG or EMG data. When the EEG or EMG acquisition system starts, it needs a TTL signal to the fiber photometry system to mark the event. In the software, the fluorescence data before this marker can be deleted, allowing the EEG or EMG and fluorescence to be accurately synchronized. Yeah. And the next question is, it is necessary to keep the experimental protected from light throughout the entire fiber photometry recording process. Yeah, the answer is please avoid the strong light during fiber photometry experiments and use the black ceramic cannulas and the black ceramic sleeves. Yeah. And also maybe you can coat the dental cement on the animal's head black to reduce ambient light interference. Yeah. So those were the questions we collected before the webinar. So now I will pick a few questions from the chat. Yeah, let me take a look at questions in the chat. Please give me a second to go through them. So the first question is, I have experience using RWD multi-channel system, and sometimes I can see fluctuations on the red 550 nanometer channel while it is actually GCAM. It is artifacts or, yeah. Yes. So yeah, the answer is we use a time division setup with two cameras, one for red and one for green. So when the red channel camera is working, it only captures red fluorescence and won't record the GCAM signal. Yeah. So you might want to check whether you are seeing its actual noise. A simple way to do that is to record the 410 channel at the same time and see if the same trend also appears there. Yeah, just wait a second, I will choose another question. We currently We're currently using the R801, R21 tricolor fiber photometry system in our lab and have been satisfied with its performance. So far, GCAM recording has been very robust and stable. I have a question regarding optimization and dual color recordings. Yeah. So thanks for your question. You raised four in total, so we will first on answering one or two of them. Sorry. Yeah. The next is, I have a question regarding recommended raw signal range and the optimization during acquisition. What is the typical recommended range of raw fluorescent signals, baseline level during recording? Yeah. If a signal appears too low or noise, which parameters would you recommend adjusting first among LED, gain, frame rate, exposure time? Do you have a preferred order or strategy for optimize these parameters? Yeah. 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 So the raw fluorescence signal can be influenced by many factors, such as the optic fiber and probe expression levels. So it's not a value that can be directly compared. Fiber photometry mainly looks at the changes in fluorescence over time. We recommend focusing on the actual output power at the fiber tip and keep it consistent across the same experimental paradigm. Yeah. So we suggest first we checking whether the exposure has been set to the maximum. You can also try lower the frame rate, which helps increase the exposure per frame. And if that still doesn't help, you may consider adjusting the power at the fiber tip, changing the gain won't improve the signal to noise ratio, so we don't recommend modifying it. Yeah, so actually, due to the time limit, yeah, so the Q&A part ends here. So if we don't get your questions today, please don't worry. You can still leave your contact information, and we will follow up with you after the session. And today's session focuses mainly on the theory and workflow. If you are interested in hands-on training, we can also organize on-site workshop tailored to your needs. We also already conducted multiple workshops in different regions, and we will be happy to support your team locally. And thank you, everyone. I hope you have a nice day. See you next time. Bye.
Against all these threats and challenges to life, constant progress of research in life science and tight collaboration in relevant industry chains are essential for achieving better vital quality. Originated in 2002, RWD has been constantly furnishing customers with reliable solutions and services in life science, animal health and clinical medicine, and dedicated for better vital quality within our wits and powers. Our products have been sold to over 100 countries and regions around the globe and won worldwide customer confidence and support. At RWD, we launched 6 solutions in the fields of life sciences, animal health, and clinical medicine at the forefront of the world, including animal surgery and modeling solutions, which ensure experiment efficiency through diverse animal disease modeling and model evaluation techniques. In vivo imaging solutions, which provide tangible tech support in fields such as biology, pharmacology, materiology, and translational medicine, among others, by accurately showing blood flow and changes in other indicators of live beings in visualized imaging. Neurosignal research solutions, which are valuable tools for neurocircuit analysis, as well as research into mechanisms for neural system diseases, development of brain-machine intelligence technology, and more. Histopathology solutions, which improve the quality and efficiency of pathological diagnosis via sectioning with high accuracy and efficiency. Cellular and molecular biology research solutions, which advance efficient scientific research through covering all conventional experiments, including cell separation and purification, self-culturing, self-analysis, and molecular biology. Animal diagnosis and treatment solutions, which evaluate diagnostic efficiency and treatment efficacy of surgery with anesthesia, dental services, rehabilitation, and physiotherapy, urinary system diseases, etc. And our assistants, science researchers, have published thousands of papers in a number of world-leading academic journals, including Nature and Science. On top of that, many scientific research institutions, medical institutions, and pet diagnosis and treatment centers have attained their success by leveraging our 6 solutions up to now. We will maintain cooperation and innovation with an open mind, boosting growth within many industries through joint efforts with our partners.
So we've been collaborating and cooperating with RWD for quite some time now, and their self-developed products have helped us in the laboratory maintain accuracy and improve accuracy and help our efficiency. We've used everything from cryostats for sectioning various organs of the body, all the way through to stereotactic surgery solutions to deliver small amounts of virus to very minute regions of the brain. We're really happy with RWD's products, and we're really excited to see what they've got in store, their future products that they have in store for the future, and helping researchers worldwide.
Mein Name ist Dr. Heinrich Kroddebusch. Ich bin seit 30 Jahren praktizierender Tierarzt und seit 20 Jahren beschäftigen wir uns schwerpunktmäßig mit der Narkosetechnik in der Kleintierpraxis. Uns Tierärzte treibt das Wohlergehen unserer Patienten an, sowie die ständige Verbesserung ihrer Lebenserwartung und ihrer Lebensqualität. RWD hilft uns dabei mit hochmoderner Beatmungstechnik und ebenso modernen Lösungen für Diagnostik und Therapie. RWDs Hilfe können wir Tierärzte sicherer und effizienter operieren, aber gleichzeitig sorgt RWD auch für eine verbesserte Lebenserwartung. Das Wohlergehen unserer Patienten und die ständige Verbesserung ihrer Lebenserwartung und Lebensdauer treibt uns Tierärzte an.
As a national high-tech enterprise, RWD ascribes great importance to technology research and development. Based on 4 technical platforms, including optical imaging, precision transmission, accurate temperature control, and detection of weak signal, we have a grip on leading core technologies across many fields. Since RWD's founding, the company has invested hundreds of millions on research and development, gaining almost 100 patents, both domestic and international, as a result. In addition to a sound manufacturing base, advanced processing assembly line, and precision detection system, RWD has built an integrated control system that monitors all aspects of operations from research and development to manufacturing. The name RWD comes from the transliteration of the English word reward, meaning payback to society. Reward is not only the original aspiration of the creation of RWD, but also the continuous belief of the company. In 2016, RWD launched the RWD Preclinical Medicine Enlightenment Scholarship to help with the development of research talent in China's preclinical medicine. So far, the scholarship has been awarded to 38 institutions of higher education nationwide. As the Vice President Unit of the Huxley Wildlife Conservation Society, RWD has donated medical equipment and supplies for wildlife relief and the conservation of the natural ecosystem. In 2020, RWD donated clinical medical supplies worth over RMB 1 million to the Giant Panda National Park to help with safeguarding our national treasure. In the future, RWD will continue to contribute its wits and powers to enhance vital quality and help global customers unlock vital mystery and safeguard vital health.